Welcome everyone. We're going to start a new chapter from plant physiology from 11th NCERT textbook, that is respiration in plants, which is chapter number 14. It is respiration in plants. Now let us see what is respiration and how it is important for plants. Now in the starting of the chapter, the author has asked us some questions. I kept the questions on the board. Let us try to solve these questions and then we will get into the chapter. The first question what the NCERT asks us is, why is breathing essential for life? Why is breathing essential for life? When we or plant or an animal, when we breathe, so what do we take? Oxygen. Oxygen is important for life. Why is breathing essential for life? The first answer we can tell like oxygen is required. Oxygen is taken through respiration, right? Or through breathing. So this is a first question answer. Then if we see the next one, what happens when we breathe? What happens when we breathe? The same thing again. When we breathe, we take oxygen in and we release carbon dioxide out. So the second question answer, the first question answer, why is breathing essential for life? Through breathing, we take oxygen inside. Oxygen is essential for life. Second one, what happens when we breathe? When we breathe, we take oxygen in and we release carbon dioxide out. So what happens when we breathe? We take, or plants take, we can tell because it is plant physiology chapter. So we can tell plants take oxygen in and they release CO2 out. Then, do all living organisms including plants and microorganisms breathe? The answer is yes. All living organisms including plants also and microbes also, they breathe. So the answer for question number 3 is yes, they breathe. Now fourth question, what did they ask us? Where does energy come from? Now from where do we get energy? For example, if I am drained out with energy, what do? I go have some food and come, so then I get the energy back again. So here the answer for this question is, where does the energy come from? Plant or animal gets the energy from the food. So we get energy from food. Next question, question number 5. How do we get energy from food? So the question for this question, the answer is we get energy from food. Now the next question is, how do we get energy from the food? Now we take the food, now we have digestive system. Inside the digestive system, the food is broken down. When the food is oxidized, when the food is broken down, then we get energy. So how do we get energy from the food? By breaking the food in presence of oxygen. It's called oxidation of food. So how do we get energy from food? Means we get energy by oxidizing the food. We get energy by oxidizing the food. What do you mean by oxidizing the food? Breaking the food in presence of oxygen. Oxidizing the food means breaking food in oxygen presence, right? Next question, question number 6. Do all foods give same amount of energy? Come on, think. Do all foods give same amount of energy? So what do we take as carbohydrate as food, protein as food and fat as food? All the food give same amount of energy? No, we studied in digestion chapter. We studied that carbohydrate will give 4 kilocalories of energy, protein also will give 4 kilocalories of energy, fats they give 9 kilocalories of energy. So that means all the foods will not give same amount of energy. Do all the foods give same amount of energy? The answer is no. For example, if you take carbohydrate gives 4 kilocalories of energy. Then if we go with protein, protein also gives its 4.1 kilocalories of energy. And if we take fat, 1 gram of fat on oxidation, it gives 9 kilocalories of energy. Fat gives 9 kilocalories of energy. 1 gram of carbohydrate gives 4 kilocalories. 1 gram of protein on breakdown gives 4.1 kilocalories. 1 gram of fat gives 9 kilocalories energy. Next question, children. Question number 7. Do plants eat? Animals only will eat. Do plants eat? The answer here is no, they don't eat. Then from where do plants get energy? We eat food, so we are getting energy. We are telling plants don't eat food. Then how do they get their energy? They trap the sunlight energy. From where do they get the energy? They get the energy from sunlight is the answer. So plants get energy from sunlight is the answer. Next question. Microorganism, question number 8. Microorganisms, do they eat food? Microorganisms are animals only. Animals eat food, right? So microorganisms also, most of the microorganisms are heterotrophs. Heterotrophs means they cannot prepare their food, so they depend on other organisms for their food. So that means microorganisms, do they eat food? Yes, they eat food like other animals. I'll write the answer here itself. So do they eat food? Yes, they eat food like other animals. Children, did you understand these 8 questions which the NCERT asks us before starting the chapter respiration in plants? Now, then the author asks us, see, like there are different questions they have asked. Don't think that all these questions are irrelevant. Why are we talking about these questions? All these questions are related to respiration only. All these questions are related to respiration only. Now, what is respiration? Inhaling oxygen is respiration. Taking oxygen is respiration. Now where will that oxygen go? Into our lungs. From lungs, who will transport that oxygen to each and every cell of our body or plant body? For us it is blood. Now in the plant system, there are mechanisms through which, through stomata, through lenticels, they will exchange their respiratory gases, right? So then that oxygen goes and reaches each and every part of the cell. Now the cell is having oxygen, the cell is also having the food. In the presence of oxygen, the cell will break the food. Breaking the food in the presence of oxygen is called oxidation. When you oxidize the food, you get energy, let it be plant or animal. So all these questions are interlinked to respiration only. Okay, take a screenshot, we will continue. Right? So we can tell that living organisms need energy and they are getting their energy by respiration. What is respiration? Respiration means taking oxygen in, supplying that oxygen to the cells. Cell will use that oxygen to break the food. When we break the food, we get energy, right? So we can tell that all the energy required for life processes comes by oxidation of food. So all the energy required for life processes, right? From where does it comes? Comes from oxidation of food. Breaking the food in the presence of oxygen is called oxidation. All the energy which is required for life comes by breakage of the food. Now, if we see, only green plants are the producers, right? Only green plants and cyanobacteria, blue-green algae, they are the only organisms on the earth which can manufacture their own food by a process called photosynthesis. So we can tell only green plants and cyanobacteria, means blue-green algae, can manufacture, only green plants and cyanobacteria can manufacture, they can manufacture their own food. By which process, ma? By photosynthesis. by photosynthesis. Now, what do you mean by photosynthesis? Photosynthesis means trapping sunlight, converting that light energy into chemical energy. So, in photosynthesis, what do? They trap light energy and they convert the light energy into chemical energy. Chemical energy means ATP energy. We know ATP is a currency, and they store it in the form of bonds. Through photosynthesis, what do plants prepare? Carbohydrate, glucose, sucrose. So they prepare chemical energy and they store it in the bonds, and they store the energy. They are storing the energy in bonds. Like, which bonds do they store? Like carbohydrates, bonds of carbohydrates. Now, what are the carbohydrates? You know. What is the simplest sugar? Simplest sugar is glucose. What is a disaccharide? Sucrose. What is a reserve food material? Starch. So, it stores the energy either in the form of glucose or in the form of sucrose or in the form of starch, right? So, green plants and cyanobacteria trap the sunlight energy, convert that sunlight energy into chemical energy and store that chemical energy in these chemical forms. Now, only green parts of the plant will prepare the food. So, we know that plant has root, stem, leaf. Now, only leaves will be green in color all throughout. So, means only the green plant, only the green part of the plant can do photosynthesis. Non-green parts of the plant, let it be flower, let it be stem, woody stem, or let it be root, which are not green, can they do photosynthesis? No. So then, like animals, like all other animals, so the non-green parts of the plants, they depend on the green parts for their food. That means only the green part. What is the green part? The green part is the leaf, and the non-green part will be the stem, and it will be the root. Only the green part will do photosynthesis, and it can manufacture food. And the non-green parts like stem and root, so they depend on the green parts for the food. That means the green part, which is called as the leaf, it has to transport the food to the stem, it has to transport the food to the root. This transport occurs through a vascular tissue called phloem. It occurs through a vascular tissue called phloem. So, transport of materials through the vascular tissue is called translocation. So, they transport the food by a process called translocation. So, we are telling that only green plants will prepare the food. Not all the part of the green plant can prepare the food. Only the green parts of the plant will prepare the food. Non-green parts like animals, they depend upon the green parts for their food only. So, we understood about plants. Now, the question is, what about animals? Animals, are they autotrophs like plants? They can prepare their food, or are they heterotrophs? Means they cannot prepare the food. The answer goes, animals are heterotrophic. Animals are heterotrophic. Heterotrophic means they cannot manufacture their food. That means they depend on plants for their food. Only plants can prepare the food. Animals are heterotrophs. They cannot prepare their food. They depend on plants for their food. Now, those animals which depend on the plant directly are called as herbivores. So, animals which depend on plants directly. Animals which depend on plants directly. So they eat grass only. What are they called? Like cow, goat, all those things. They are called herbivores. They are called herbivores. And animals which don't depend directly on the plant, right? So the cow is eating the grass and the cow is eaten by other animal, or the goat is eaten by other animal. Means they are flesh eating animals. That means they are not herbivores, they are called carnivores. So we can tell animals which depend on plant indirectly. Animals which depend on plant indirectly. What are they called? They are called carnivores. They are called carnivores. But let it be herbivore or carnivore, it is heterotroph only. Now let us talk about fungi then. So we talked about plants. Yeah, where is that? We talked about plants, we talked about cyanobacteria, we talked about animals. Now let us talk about fungi also. Now fungi are saprophytic. What are fungi? They are saprophytic. What do you mean by saprophytic mode of nutrition? Saprophytes means they obtain their food from dead and decomposing organic matter. So on the dead and decomposing organic matter, they secrete enzymes, they digest their food and take the food inside. Such a mode is called as saprophytic mode of nutrition. Fungi have saprophytic mode of nutrition. They have saprophytic mode of nutrition. What do you mean by saprophytic mode of nutrition? Obtain food from dead and decomposing organic material. So they get food from dead and decomposing organic matter. They get their food from dead and decomposing organic matter. Understood till here? So we are telling that energy is required for the life to be maintained. And how do you get energy? When you break the food, we get energy. Breaking the food in presence of oxygen is called oxidation of food. Green plants and cyanobacteria can prepare their food by a process called photosynthesis. Only the green parts of the plant will prepare the food. Non-green parts, they depend on the green parts to take their food. Now coming to animals, animals are heterotrophic. The animals which depend on plant directly are herbivores. The animals which depend on plants indirectly for the food are called carnivores. Coming to fungi, fungi are saprophytic. Saprophytic means they grow on dead and decomposing organic matter. Take a screenshot, we will continue. Right? So we can tell that all the food for the life processes comes from plants only. On the earth, what are those organisms which can manufacture the food? Means plants only and only plants. So we can tell that like all the food for the life processes comes from photosynthesis only. All food for our life processes, from where does it come? It comes from plants by a process called photosynthesis. By a process called photosynthesis only food is prepared. Now where will photosynthesis takes place? PS means photosynthesis. Photosynthesis occurs in chloroplast. Right? And where does respiration occurs? What do you mean by cellular respiration? Inside the cell, using the oxygen, breaking the food to get energy is called respiration. Respiration, where does it occur? Photosynthesis occurs in an organelle called chloroplast, and respiration occurs in an organelle called mitochondria. Now, these two are organelles. Chloroplast and mitochondria are double membrane structures, and these double membrane structures are seen only in eukaryotic cells. Mitochondria and chloroplast will be there only in eukaryotic cells. So in eukaryotic cell, food is prepared. In chloroplast, food is broken down in mitochondria. When we break the food, when we do respiration in the mitochondria, so what happens? Energy, ATP is released or ATP is generated. ATP is made, right? So now if we have to ask what is respiration? Respiration means breaking the food, organic material means which is having CC bonds. We are breaking the bonds. So breaking the CC bonds through oxidation. So respiration includes Respiration, what does it includes? It includes breaking the CC bonds. Breaking CC bonds. Breaking the bond in presence of oxygen is called oxidation. Breaking the bonds through oxidation. When we break the bond through oxidation, it leads to release of energy. Breaks the bonds through oxidation, leading to, we have seen that energy is released, leading to release of energy. Energy will be released. This process is called as, what is this process called? Is called as respiration. Where does the respiration takes place? Inside the cell. So we call it as cellular respiration. We call it as cellular respiration. Did you understand, children, what is cellular respiration? Cellular respiration means it is breaking. What are you breaking? You're breaking organic compounds. Which bonds are you breaking? CC bonds. In whose presence are you breaking? In the presence of oxygen. What are you getting? Energy. What is this process called? Respiration, right? Now the compounds which are used for breakdown are called as respiratory substrate. The person who teaches is called as a teacher. Like that, the compounds which are oxidized, which are broken, are called as respiratory substrates. So the compounds which are oxidized, the compounds which are oxidized, what are they called? They are called respiratory substrates. The compounds which are broken are called as respiratory substrates, right? Now, in most of the cases, who is a respiratory substrate? Usually carbohydrates like glucose, sucrose, starch are the respiratory substrates, right? Usually carbohydrates are the respiratory substrates. Now, when we are eating the food, we take starch, right? But then when we are starving from past 2 days, if you don't eat anything, then from your muscle protein, we get food, we get energy, or from the glycogen, we get energy, or from the fats, we get energy. So that means usually carbohydrate is the respiratory substrate, but proteins also, fats also, in plants organic acids are also the respiratory substrates. So we can tell usually carbohydrates are the respiratory substrates. Usually carbohydrates are the respiratory substrates, but we can tell proteins, fats and organic acids are also the respiratory substrates. Not only carbohydrates, proteins, fats and organic acids are also respiratory substrates. Understood? Now, during oxidation, all the energy which is present in these respiratory substances, is it released free into the cell? Where is respiration happening? Respiration is happening in a cell. That too where? Inside the mitochondria. So when you are breaking the food, is all the energy liberated into the cell? If all the energy is liberated into the cell in the form of heat energy, then the heat will get dissipated off, distributed off, wasted off. So that means, so when you are breaking the respiratory substrates, energy is not released into the cell directly. Energy is not released as heat energy, or we can tell energy is not released in one step. Huge amount of energy will not be released in one step, right? We can tell that slowly, step by step, enzymatically. Enzymes means the reaction can be controlled. Slowly, controlled by enzymes, the respiratory substrates are broken step by step to release energy, and that energy will be coupled with ATP synthesis. That energy will be coupled with ATP synthesis. What is ATP? The currency of the cell. That ATP will be stored in the cell. Whenever the cell needs energy, then it will take the ATP, it will break the ATP, then it will get the energy. So take a screenshot, we will continue. right? So what are we telling? All the energy present in the respiratory substrates is not released into the cell. We are telling it's not released into the cell directly. So we are telling it is released slowly. Energy is released slowly, and energy is released in many steps, and the breakdown is controlled. The breakdown is controlled by enzymes, so that means it is under control, and the energy which is released is not released as heat energy. Energy which is released is coupled to ATP synthesis. The energy is coupled to ATP synthesis. So 1, 2, 3, when you are breaking these bonds, CC bonds, when we are breaking, we are getting energy. Now this energy, if it comes out like this, it becomes heat energy, and that heat energy will be dissipated off, will be distributed off. So the plant or the organism is very clever. It is not releasing the energy like this, it is coupling this. Means it uses this energy to combine ADP with inorganic phosphate to make ATP. It will make ATP. So ATP synthesis will happen when you break the food. Now that is why ATP is called as currency of the cell. It's called as the currency of the cell. As and when cell needs the energy, cell will break the ATP and gets its energy. Understood, children? Understood till here? Now, in the NCERT, the next topic is 14.1. The point is, do plants breathe? They are asking us, do plants breathe? Come on, tell me, do plants breathe? Yes, plants are living organisms. We told that all living organisms breathe. They do respiration. So do plants breathe? So the answer is yes, plants breathe. Yes, like all other animals, plants also breathe. They also breathe. What are they doing during breathing? They are taking oxygen in, they are releasing carbon dioxide out. So what do? They consume oxygen and they release carbon dioxide like animals. Plants also, like animals, when they are doing respiration, they take oxygen, they release carbon dioxide. So we have respiratory system starting from the nostrils till the lungs. We have respiratory system to tackle the needs of respiration. Like that, the author asks a question, like plants, do they have specialized respiratory organs like lungs and all? No. So we can tell plants, they breathe, but they do not have specialized respiratory organs. They breathe, but they don't have specialized respiratory organs. And the author tells that plants can survive without respiratory organs because of 3 reasons. So plants are doing respiration, but they don't need respiratory organs. What are the explanations they have given? Means they have given 3 points, 1, 2 and 3 points. Let us see what they have given here. The first point, what they tell is, each part of the plant take care of its own gaseous needs. So that means, like our lungs, who has to take oxygen in and who is taking the role of distributing that oxygen through blood to all the parts of our body. Not like that in the case of plant. In plant, each part take care of its own gaseous need. Root will take care of its own respiration, stem will take care of its own respiration, leaf will take care of its own respiration. So then when each part is taking care of its own respiration, specialized system is not required. That's what they tell. So each part take care of its own gaseous exchange. Each part of the plant take care of its own gaseous exchange. That means very little gaseous exchange occur from one organ to other organ. So very little gaseous exchange occurs from one part to other. This is the first explanation the author will give why plants doesn't require any respiratory organs. Next point, they tell that plants do not have great demands of respiratory gases like animals. Plants, where they are only they are. They don't move from their places, right? So when they don't move, they don't do much respiration. So that's the other point. Plants do not have great demands of gases exchange like animals. So plants do not have great demands of gaseous exchange like animals. Why, children? Why? Because they are static. Because the plants are static, they don't move. Only during photosynthesis they do more gases exchange. In photosynthesis, what do? They take carbon dioxide in, they release oxygen out. So only during photosynthesis large volumes of gases are exchanged, right? We are telling plants don't have great demands of gases exchange, but only during photosynthesis. Only during photosynthesis great volumes of gases are exchanged. Great volumes of gases are exchanged. So when they do photosynthesis, they take CO2 in and they release oxygen out. That oxygen, what they want, they will use that oxygen for themselves. The remaining oxygen only they will release it outside. No. So during photosynthesis they need not do respiration because oxygen is already there in their cells. That's the second explanation. And third point, they will tell that the distance that gases must diffuse even in tall plants is not huge. So the third point, they'll tell the distance that gases must diffuse even in tall plants is not huge. Plants are very much tall than us. So even in tall plants also, the distance what gases are diffusing are not that huge when compared to animals, because each part of the plant somehow it is in contact with the outside atmosphere, right? They have openings, through stomata, through lenticels, or through loosely packed parenchymatous cells. Even the inner parts of the plant are also in connection with the outer atmosphere. How are they in contact with the outer atmosphere? Through their stomata or their lenticels or by loosely packed parenchyma, or by loosely packed parenchyma, they are in contact with their surrounding atmosphere, so they can take the respiratory gases directly. So with these 3 points we can conclude, we can tell that plants can survive without specialized respiratory organs. Take a screenshot, we will continue. Right, so now we tell that when we do combustion of the glucose in the presence of oxygen, so means combustion means breakdown. So what do we get? The complete combustion means completely breaking all the CC bonds of the glucose. So if you see 1, 2, 3, 4, 5, 6. This is glucose, right? So when you break this bond, this bond, this bond, this bond, this bond, right? So it is aldehyde, this is glucose. So when you break all the CC bond, CC bond, CC bond, CC bond, so then what do we get? So remove these hydrogen, Sama. take off the hydrogens, when you take off the hydrogens, only C and O, C and O, C and O, C and O will be there. So that means glucose is broken down. This is glucose. What do you mean by glucose? C6H12O6 is broken down in the presence of oxygen. 6 O2 molecules. When you break glucose in the presence of oxygen, what is the process called? Oxidation of food. So when we oxidize the food, so when we break the bonds, what do we get? So 1, 2, 3, 4, 5, 6 CO2s. We get 6 CO2s, carbon dioxide molecules. How do you get? By removing hydrogens we are getting. Who will take these hydrogens? Oxygen will take the hydrogens. After taking hydrogens, oxygen will become 6 water molecules. And we know when we are breaking anything, energy will be released out. So breakdown process, respiration is a catabolic process. Catabolic process means it is a breakdown process. So when we break anything, energy will be released. Energy releasing process are called exergonic processes, right? Or exothermic processes. It is called exothermic process. That means we are telling energy is released out. Energy is released out. So when you do combustion, the energy will be in the form of heat energy. When you do outside, when you burn a compound outside, so then we call it as combustion. In combustion, energy will be released out as heat energy, but that heat energy will be lost, will be dissipated out, will be distributed out, right? The heat energy will be lost. So that is why now living organisms like plant or animal, what do? They don't release heat energy. Instead, they release chemical energy. They convert it into chemical energy. What chemical energy do they make? They convert it into adenosine triphosphate, ATP molecule. What is ATP? It is the currency of the cell. ATP cannot be wasted like heat energy. So very smart, right? Living organisms are. So when they are breaking anything, they are getting energy. That energy, they know if they release it into heat energy, then that heat energy will be lost. So that is why it will release only little amount of heat energy. Remaining energy, it will convert it into chemical energy like ATP. Understood till here? Now we understood that respiration means oxidation of food, breakdown of the food. Now take a screenshot, we will continue. Now what are the steps of respiration? Let us see. Okay. Now there are two types of respiration depending on whether oxygen is required or oxygen is not required. So depending on oxygen requirement. So respiration is of two types. One is called aerobic respiration and the second one is called anaerobic respiration. So the name itself tells ana means no oxygen. Anaerobic respiration means in absence of oxygen. So anaerobic respiration means in absence of O2. Aerobic respiration means in presence of O2. Now, if you ask me to take the class by closing my nose, so can I teach? No, I can't. So that means if you do respiration without oxygen, so then will it be able to do complete oxidation? Will it be able to break all the bonds? No. So that means anaerobic respiration, is it complete breakdown? I told no. So that means it is partial breakdown. So we can tell in anaerobic respiration, like the substrates are partially oxidized. They are not completely broken, they are partially oxidized. Whereas in aerobic respiration, the substrates are completely oxidized. The substrates are completely oxidized. Now, just now we saw the structure of glucose, and when we tried to broke all the CC bonds, CC bonds, CC bonds, CC bonds, what do you get? When you break, when you completely oxidize, we get 6 molecules of carbon dioxide and 6 molecules of water, right? So, in aerobic respiration, the end products are carbon dioxide plus water. Right? So when we break it completely, CC, when we remove those hydrogens, you will get carbon dioxides and water, 6 CO2s and 6 water molecules we get. When we do anaerobic respiration, we are not breaking all the bonds. So then we don't get carbon dioxide and water. Now, if it is, then anaerobic respiration, it is called as fermentation also. It is called as fermentation also. If it is ethanol fermentation, if it is alcohol fermentation, then what do we get? If it is acid fermentation, what do we get? In alcohol fermentation, we get C2H5OH plus carbon dioxide. Try to break one glucose molecule. What is glucose? 1, 2, 3, 4, 5, 6. So you need to get alcohol, right? So that C2, that means this is one alcohol and this is another alcohol. So 2 alcohols you will get, and this is CO2, this is CO2. So 2 alcohols and 2 CO2s you will get. In alcohol fermentation, we get 2 alcohols and we get 2 CO2s. And where do you find alcohol fermentation? Means in yeast. During, during alcohol preparation, during bread preparation, right? So we see this process. Whereas acid fermentation, lactobacilli, right? In lactobacilli, where do we see this? We see this in lactic acid bacteria. And even in our muscles also, when we are doing exercises. Even in our muscles also, when we are doing exercises, then what happens? The glucose is broken down to lactic acid. So 1, 2, 3, 4, 5, 6. It's broken down to one lactic acid, another lactic acid. See, we are not breaking all the CC bonds. In lactic acid also, this CC bond is there, this CC bond is there, this CC bond is there, this CC bond is there. You have broken only this CC bond. So either in lactic acid formation or in alcohol formation, CC bonds are still there. So means it is partial oxidation only. So in acid fermentation, what do we get? We get lactic acid only. Is carbon dioxide forming? No. Only in alcohol carbon dioxide comes. But in acid, since it is a 3 carbon compound, no CO2 is evolved. In lactic acid formation, no CO2. This came in NEET examination. In lactic acid formation, is carbon dioxide is involved or not involved? This came in NEET examination, NEET paper. Okay, so we are seeing the differences between aerobic respiration and anaerobic respiration. In aerobic respiration, oxygen is required. In anaerobic respiration, oxygen is not required. In the presence of oxygen, the substrate, the glucose molecule is completely broken down into carbon dioxide and water. In absence of oxygen, the glucose is partially broken down either into alcohol or into lactic acid. Now, when we are breaking, we are telling we are getting energy. When you break all the bonds, we get more amount of energy here. When you break only 1 or 2 bonds, we get very little amount of energy here. So, in terms of energy, we can tell that complete breakdown yields 38 ATP molecules. Isn't it very huge? 38 ATP molecules will be obtained when you completely break the glucose molecule. Whereas in anaerobic respiration, right, only 2 ATP molecules are obtained. Since it is half the way, since it is partially oxidation, so it gives just 2 ATPs only. So that's why they'll tell, if you start any process, if you want to get the complete benefits from it, go to the last. Do it till the end. Don't leave it in between, so that will not be fetched with anything. See here, it has broken incompletely. It's just getting 2 ATP molecules. But when you complete the process, you'll get 38 ATP molecules. Okay. Now, aerobic respiration has how many steps? Anaerobic respiration has how many steps? Aerobic respiration has 3 steps. 1, 2, 3. The first step of breaking glucose is called glycolysis. Lysis of glucose is the first step in aerobic respiration. Then goes with TCA cycle. We'll see what is TCA cycle. Then goes with electron transport system. The 3 steps of aerobic respiration are glycolysis, TCA cycle and electron transport system. Whereas the steps of anaerobic respiration are, one is glycolysis and the other one is fermentation. One is glycolysis and fermentation only. So that means glycolysis is common here also, here also. So since it is common in both the places, we tell it is core respiration. Core respiration means glycolysis, which is common during aerobic respiration and anaerobic respiration. And where can you find aerobic respiration? In all eukaryotic higher plants and animals. Where can we find aerobic respiration? In plants and animals we find aerobic respiration. So these are the differences between aerobic and anaerobic respiration. Take a screenshot, then we will start with the first step of respiration, that is glycolysis. Take a screenshot. The 3 steps of aerobic respiration are, 1 is glycolysis, the second one is TCA cycle, and the third one is electron transport system, right? So we'll start with glycolysis, which is part of aerobic respiration. Let us start with glycolysis now. When we see glycolysis, the word glycolysis is derived from a Greek word. It is derived from a Greek word in which glucose means sugar and lysis means breakdown. Right? I think we understood what is the meaning of glycolysis. It is derived from a Greek word. Glucose means sugar, lysis means breakdown or splitting up. And this glycolysis, where does it occur? Means location. It happens in the cytoplasm of the cell. It occurs in the cytoplasm of the cell. And who elucidated the pathway of glycolysis? 3 scientists. Their names are, one is Emden, second one is Meyerhoff, and third one is Paranas. They elucidated the pathway. So that is why after their names it is called as EMP pathway. So why glycolysis is also called EMP pathway? After the names of the scientist. What are the names of the scientist? The first scientist name is Gustav Emden. And the second scientist name is Otto Meyerhoff. And the third scientist name is J. Paranas. After these scientists' name, it is called as EMP MP pathway, Embden-Meerhoff-Paranas pathway, right? So now what happens in glycolysis means it is the process of breakdown of glucose, partial oxidation of glucose. So glucose, which is a 6-carbon compound, is broken down into a 3-carbon acid which is called as pyruvic acid. What happens in glycolysis? In this process, glucose is partially oxidized. Why are we telling it is partially oxidized? Because it is not completely oxidized into carbon dioxide. It is oxidized into 3-carbon compound called as pyruvic acid. It is partially oxidized into 2 molecules of pyruvic acid. 2 molecules of pyruvate. Means glycolysis starts with glucose. Means who is the substrate? The substrate is glucose molecule. And what is the product of glycolysis? We are breaking the 6-carbon compound into 2 3-carbon compounds. What are the 3-carbon compounds? That is a product. It is called as pyruvate. What is it called? It is called pyruvate, or we can call it as pyruvic acid. It is also called pyruvic acid. So, understood, children? We will see now the steps of glycolysis. Take the screenshot of this. Right? Now, glycolysis is a process. It is a 10-step reaction. So, we will see step by step now clearly. Okay? We'll first start like the product of photosynthate. Plant will do photosynthesis, and what is the product of photosynthesis in the plant? The product of photosynthesis in the plant is sucrose. Now, what is sucrose? Sucrose is a disaccharide made up of 2 sugars. We need to convert the disaccharide into a simple sugar called monosaccharide. Now, we'll try to break the sucrose with an enzyme called invertase. When we break the sucrose with an enzyme called invertase, then what do we get? So, we get 1 glucose molecule and 1 fructose molecule. Sucrose, which is a disaccharide, it is broken down with the presence of an enzyme called invertase into glucose and fructose. Glucose is a 6-carbon aldose sugar. Fructose is a 6-carbon ketose sugar. Now, glucose and fructose, both of them will enter glycolysis. Now, we will start glycolysis. Glucose and fructose, what do? They start up the first step. What is the first step called? The first step is called phosphorylation. It is called as phosphorylation of the sugar. Means we need to add phosphate to the sugar. Means who will come to donate the phosphate? ATP has 3 phosphates. It will donate the phosphate and it will become diphosphate. So who will come to donate the phosphate? We are bringing ATP, will donate the phosphate and it becomes ADP. So after taking this phosphate, glucose will become glucose phosphate. Where is that phosphate added? At the 6th place of the glucose. So glucose and fructose also gets converted to glucose 6-phosphate. It gets converted to glucose 6-phosphate, right? This is the first reaction, which is called as phosphorylation. So the name of the enzyme is called as hexokinase. The name of the enzyme is called hexokinase. Why is it called hexokinase? Because hexose is getting phosphorylated glucose or fructose is getting phosphorylated enzyme is called phosphorylase or kinase. So the reaction phosphorylation, we got hexokinase is the first reaction. Now what happens in the second reaction? In the second reaction, glucose 6-phosphate wants to get converted to fructose 6-phosphate. Glucose 6-phosphate wants to get converted to fructose 6-phosphate. Means this reaction is called isomerization. Glucose and fructose are functional isomers. So the name of the reaction is called isomerization. The enzyme is called as glucose phosphate isomerase. What's the name of the enzyme? Glucophosphoisomerase is an enzyme. So children, the first reaction is glucose getting converted to glucose 6-phosphate. Second reaction is glucose 6-phosphate getting converted to fructose 6-phosphate. The reaction is isomerization. Now third reaction is fructose 6-phosphate wants one more phosphate. It is demanding for one more phosphate. Now again, whenever phosphate it needs, who will come to donate the phosphate? ATP will come to donate the phosphate. So here the ATP molecule will come. After giving off the phosphate to fructose, ATP becomes ADP. Then the fructose, which is already having phosphate at the 6th place, is taking an extra phosphate at the 1st place. So then it becomes fructose 1,6 diphosphate or bisphosphate. Fructose 1,6 bisphosphate is a reaction. So reaction number 3, it is again phosphorylation of the sugar. So reaction 3 is called as phosphorylation of the sugar. The name of the enzyme is called as fructose phosphokinase or phosphofructokinase. What is the name of the enzyme? The name of the enzyme is fructophosphokinase is an enzyme, right? So we got an energetic molecule which is called as fructose 1,6 bisphosphate. Now it is ready to break. Now it is ready to break. So take a screenshot. We will continue the fourth reaction on the fresh board. Right. So we'll start with fructose 1,6 bisphosphate. So glucose got converted to glucose 6-phosphate, glucose 6-phosphate got converted to fructose 6-phosphate, fructose 6-phosphate got converted to fructose 1,6 bisphosphate. Now reaction number 4, fructose 1,6 bisphosphate is breaking up. Fructose 1,6 bisphosphate is breaking up. So it's a breakdown reaction. So the name of the reaction is called cleavage. The name of the enzyme which helps in cleavage is called aldolase. So when you are breaking a 6-carbon sugar, you will get 2 3-carbon sugar. So one sugar is called as aldotriose. The other sugar is called as ketotriose. So when you are breaking a 6-carbon sugar, it's obvious that you will get 2 3-carbon compounds. One 3-carbon sugar is called triose. One 3-carbon sugar which is having aldehyde as a functional group is called aldotriose. Another 3-carbon sugar which is having ketose as a functional group is called ketotriose. The name of the ketotriose is DHAP. The name of the keto sugar is called DHAP, dihydroxyacetone phosphate. What is the name of the keto sugar? Dihydroxyacetone phosphate is the name of the keto sugar. Now what is the name of the aldose sugar? We know aldotriose is glyceraldehyde phosphate at the third place. It is called as glyceraldehyde 3-phosphate. In NCERT they have given the short form as PGAL, phosphoglyceraldehyde. So we can write it as phospho 3-phosphate, third place phosphate group added, 3-phosphoglyceraldehyde is aldotriose. This is ketotriose, so it has broken down. Reaction number 4. Now fifth reaction is, what is fifth reaction? In fifth reaction, the ketotriose, this is reaction number 5. The ketotriose gets converted to aldotriose. The ketotriose is getting converted to aldotriose. Then that is reaction number 5. It is triose isomerization. The triose is getting isomerized. So reaction number 5, I'm writing it down here. It is called triose isomerization. And the name of the enzyme will be isomerase, triose isomerase. The name of the enzyme will be triose isomerase is the name of the enzyme. So already one PGAL is there. DHAP also got converted to PGAL. Then we are getting 2 molecules of PGAL. So write down, we are having 2 molecules of PGAL. How do you get 2 molecules of PGAL? One PGAL directly from fructose it is formed. After isomerization, this also got converted to this. So we got 2 molecules of PGAL, phosphoglyceraldehyde. Now let's do the 6th reaction. Now reaction number 6. In 6th reaction, what happens means phosphoglyceraldehyde. Phosphoglyceraldehyde gets converted to glyceric acid. Aldehyde getting converted to glyceric acid. So then it gets converted to 1,3-bis phosphoglyceric acid. So phosphoglyceraldehyde got converted to glyceric acid, one change. It took one more phosphate, another change. So it has taken one phosphate, first point, and it has lost hydrogen atoms. It has lost hydrogen atoms. Those hydrogen atoms are taken by NAD+. So 2 molecules will come for 2 phosphoglyceraldehydes and it will become 2 molecules of NADHH+. From glyceraldehyde, 2 hydrogen molecules are taken by NAD and it gets converted to 2 molecules of NADHH+. And phosphate gets added up at the first place. So we are getting 1,3-bis phosphoglyceric acid. How many molecules? 2 molecules. So 2 molecules of 1,3-bis phosphoglyceric acid are formed. Removal of hydrogen is called oxidation. So reaction number 6 is called as, what is this called as? It is called as oxidation. First time we are seeing oxidation. So it is called oxidation 1. Wherever NADHH+ is made, the reaction will be called oxidation. The name of the enzyme will be called dehydrogenase. So you removed hydrogens from glyceraldehyde. So the enzyme will be called as, in shortcut, we can call it as GDH. It's called GDH, glyceraldehyde dehydrogenase. What is the name of the enzyme? The name of the enzyme is, from glyceraldehyde we removed hydrogen, so it is called as glyceraldehyde dehydrogenase. I'll write the full form here. Glyceraldehyde dehydrogenase is an enzyme. So this is reaction number 6. Now let us do the 7th reaction. In 7th reaction, 1,3-bis phosphoglyceric acid is loaded with phosphate. It wants to donate phosphate now. So when it is ready to leave the phosphate, who will come to take the phosphate? ADP. So it is giving the phosphate. ADP will take the phosphate and it will become ATP. For 2 molecules, 2 ADPs will make 2 ATP molecules. ATP is made here. So that means the reaction will be phosphorylation. The phosphate is given by the substrate molecule here. So it is called as substrate level phosphorylation. Reaction number 7. It's to be called as substrate level phosphorylation 1. Substrate level phosphorylation 1. 2 ATP molecules are made. Wherever ATP is made, the enzyme will be called as kinase. So it is called as phosphoglycerokinase. What is the name of the enzyme? The name of the enzyme is called phosphoglycerokinase is the enzyme. Phosphoglycerokinase is an enzyme. After giving off the phosphate, it will become 3-phosphoglyceric acid. Means it has lost the phosphate from the first place. So it becomes 3-PGA. We will write it as, we can write in short form as 3-PGA, 3-phosphoglyceric acid. Now let us do the 8th reaction also. 3-PGA, 3-phosphoglyceric acid gets converted to 2-phosphoglyceric acid. 3-phosphoglyceric acid gets converted to 2-phosphoglyceric acid, means the phosphate which is at the third place has shifted to the second place. So that means the reaction is called as intramolecular shift, shifting of phosphate from the third place to the second place. So the reaction is called as intramolecular shift. Reaction is called intramolecular shift, and the enzyme, it's something like mutation, so it is called mutase. The enzyme is mutase. 3-PGA got converted to 2-phosphoglyceric acid. How many molecules? 2 molecules. So 2 molecules of 2-phosphoglyceric acid are formed. So first reaction is conversion of glucose to glucose 6-phosphate. Second reaction is conversion of glucose 6-phosphate to fructose 6-phosphate. Third reaction, conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Fourth reaction, cleaving the fructose into aldotriose and ketotriose, cleavage. Fifth reaction, converting this triose into this triose, so it is isomerization. Sixth reaction, oxidation 1, where NADH plus is formed. Seventh reaction is phosphorylation 1, substrate level phosphorylation 1. Eighth reaction is intramolecular shift, enzyme mutase. We got 2 molecules of PGA. Take a screenshot, we will continue. Right. So now we are with 2 molecules of PGA. Excuse me. So 2 molecules of 2-phosphoglyceric acid. Now what is this? This is phosphoglyceric acid. Now 9th reaction, we will convert phosphoglyceric acid into phosphoenol pyruvate. So phosphoglyceric acid after losing water molecule, so it is dehydration reaction. Since water molecules are coming out, the reaction is called dehydration reaction. The name of the enzyme will be called as enolase. What is the name of the enzyme? It is called enolase. So then phosphoglyceric acid gets converted to phosphoenol pyruvate. How many molecules? 2 molecules of phosphoenol pyruvate. As I told you, glycolysis is a 10-step process. So the last step is conversion of phosphoenol pyruvate to pyruvate. Conversion of phosphoenol pyruvate to pyruvate. How many molecules? 2 molecules of pyruvic acid are formed. Means what did we do here? We removed phosphate. Who will come to take the phosphate? ADP will come to take the phosphate. After taking the phosphate, it will become ATP. ADP got converted to ATP. What is this reaction? Phosphorylation. Substrate is giving phosphate. So the 10th reaction is called as substrate level phosphorylation. Substrate level phosphorylation 2. Substrate level phosphorylation 1 we have seen in the 7th reaction. This is substrate level phosphorylation 2. Enzyme will be called as pyruvate kinase. What is the name of the last enzyme? It is pyruvate kinase. The last enzyme is called pyruvate kinase. This is what is glycolysis. In glycolysis, it's partial oxidation of glucose, which is a 6-carbon sugar, into 2 molecules of 3-carbon acid. Pyruvic acid is a 3-carbon acid into 2 molecules of 3-carbon acid, pyruvic acid we got. So, total, how many ATPs are formed? Total, how many ATPs are directly produced? They are directly produced by glycolysis. 7th reaction, 2 ATPs are made. 10th reaction, 2 more ATPs are made. So 2 plus 2, we can tell 4 ATPs are formed. But then if you recollect, in the first reaction we used 1 ATP, in the third reaction also we used 1 ATP, glucose to glucose 6-phosphate, fructose 6-phosphate to fructose 1,6-bisphosphate. Since 2 ATP molecules are used up, 4 minus 2, net is 2 only. So 2 ATPs, so 4 ATPs minus 2 ATPs. So the net will be 2 ATPs only. So by doing glycolysis, direct ATPs we are getting is 2 ATP molecules. When this NADHH plus, in the 6th reaction we got 2 molecules of NADHH plus. When you take it to aerobic respiration, then it will give more ATP. So that they did not ask us in NCERT. In NCERT they asked directly how many ATPs are made. In the 7th reaction 2 ATPs, and in the 8th reaction 2 ATPs, and in the 10th reaction 2 ATPs. 4 ATPs are made. Take a screenshot, we will continue. Right. Now, what is the end product of glycolysis? The end product of glycolysis is pyruvic acid. Now, what is the fate of pyruvate? What is the fate of pyruvate? Now, after completing your 10th standard, what is the fate? We can enter into commerce stream, we can enter into medical stream, we can enter into engineering stream. 3 routes we have. In the same manner, the fate of pyruvate, pyruvic acid, depending upon the requirement of the cell, it can take 3 routes. It can take 3 routes. What are the 3 routes? So, the first route is to continue aerobic respiration. If it has to continue aerobic respiration, we know it has to enter TCA cycle and it has to do electron transport system. So this is a lengthy story, we will handle it. Now we'll see the next 2 fates. Now the next 2 fates are called fermentation. This is also fermentation, this is also fermentation. Now this we told it will continue aerobic respiration. Fermentation means it is anaerobic respiration. So pyruvic acid under anaerobic conditions, it will continue fermentation. Now if it is yeast, what it will do? If it is lactic acid bacteria, what it will do? If it is yeast, what it will do? If it is lactic acid bacteria, what it will do? So we have seen that the in glycolysis, glucose got converted to glucose 6-phosphate, got converted to fructose 1,6-bisphosphate, right? Somehow it got converted, after some reactions it got converted to pyruvic acid. It got converted to pyruvic acid. Now this pyruvic acid, it can make alcohol also, it can make lactic acid also. This pyruvic acid in bacteria, lactic acid bacteria, which produces an enzyme LDH. LDH means lactate dehydrogenase. It produces an enzyme called lactate dehydrogenase. Then the pyruvic acid gets converted to lactic acid. it gets converted to lactic acid. This is a 3-carbon acid. This is also a 3-carbon acid. In one step it gets converted. Now for conversion of pyruvic acid to lactic acid, it needs NADHH plus molecule. So what happens? It is consuming the NADHH plus which is formed in the 6th reaction. NADHH plus is oxidized to NAD. That means hydrogens are taken from NAD given to pyruvic acid to make lactic acid. Since the substrate is taking hydrogens, the reaction is called reduction. What is this reaction called? This reaction is called reduction. Right? The reaction is called reduction. The name of the enzyme is called lactate dehydrogenase. What's the name of the enzyme? It is lactate dehydrogenase. It happens in lactic acid formation, means when milk is getting converted to curd, lactose gets converted to lactic acid in this manner, okay, where the NADHH plus molecules are used up to make a 3-carbon lactic acid. Carbon dioxide is not involved. Whereas when we talk about yeast, in yeast what happens? In yeast what happens? The pyruvic acid molecule, which is a 3-carbon compound, it gets converted, it undergoes decarboxylation. Pyruvic acid, which is a 3-carbon molecule, after losing carbon dioxide, it gets converted to a 2-carbon compound called acetaldehyde. An intermediate called acetaldehyde is formed. Then this acetaldehyde, it takes hydrogens from NADHH plus. After taking the hydrogens from NADHH plus, acetaldehyde gets converted to ethanol. Acetaldehyde gets converted to ethanol, which is 2-carbon compound. Here it is 1-step process. Here it is 2-step process. This reaction is called as decarboxylation. Since carbon dioxide is removed out, it is called decarboxylation. The enzyme is called pyruvate decarboxylase. The name of the enzyme will be pyruvate decarboxylase. Whereas the second reaction is reduction reaction, mediated by an enzyme called alcohol dehydrogenase. This second reaction is called reduction, mediated by an enzyme called ADH, stands for alcohol dehydrogenase. So in yeast, when pyruvic acid is getting converted to ethanol, which is a 2-carbon compound, CO2 is released. And it takes place in 2 steps. Acetaldehyde is an intermediate. Whereas in lactic acid bacteria and in animal muscle cells, when we are doing exercises, when the oxygen intake is not sufficient, so we are doing more exercise, the oxygen what we are taking is not sufficient, so then anaerobic conditions will build up in the muscle. So then this condition happens where pyruvic acid is reduced to lactic acid. So these two are fermentations. So in yeast it is ethanol fermentation, in lactic acid bacteria it is lactic acid fermentation. So, and if it is the third way, it will do aerobic respiration. Take a screenshot, we will continue. Now, in which organisms we can see fermentation means? In yeast and bacteria, means in unicellular eukaryotes and in prokaryotes, right? So, where can we see fermentation? Fermentation occurs in yeast, which is a unicellular, in some unicellular eukaryotes and prokaryotes like bacteria. We can see fermentation. And fermentation, anaerobic process, complete oxidation or partial oxidation. It is partial oxidation. So if it is partial oxidation, the energy yield will be very much less. In lactic acid fermentation also and alcohol fermentation also, not much energy is released. So both in lactic acid fermentation and alcohol fermentation, not much energy is released. Not much energy is released, correct? So then how much energy is released means less than 7%. Less than 7% energy is released. That's it. So we can tell less than 7% of energy of glucose is released. Okay, in terms of ATP, I have to tell it's only just 2 ATPs are released. That we calculated, right? In glycolysis we calculated and we told 2 molecules of ATP, only 2 molecules of ATP are released, right? So this is fermentation, and it makes acid, it makes alcohol. So both are hazardous, right? So we can tell the process is, fermentation process is hazardous. Fermentation process is hazardous. Both acid also and alcohol also are not good. Yeast is only making alcohol. Yeast itself will poison themselves if the alcohol content is above 13%. So yeast also poisons themselves. When it will poison themselves? When the alcohol content, when alcohol content is about 13%. If the alcohol content is 13%, then yeast will die. That means under natural fermentations, when they are making liquor, when they are making alcohol, how much percent alcohol it can make? Only 13% alcohol we can make. So what are those alcoholic beverages called? So beer and wine. So they are examples of non-distilled beverages. In non-distilled beverages, the alcohol content will be 13% only. Whereas when you concentrate it, when you distillate it, then the alcohol content increases to 30, 40, 60, 70, 80 also. Then they are called as non-distilled beverages. Examples for non-distilled beverages are vodka, brandy, rum, gin. Okay, all these things are examples of distilled beverages where the alcohol content will be more than 13%. It can be 25, it can be 50, it can be 70, it can be 80% also. So this is about fermentation, alcohol fermentation and acid fermentation. Now we understood fermentation. We'll talk about the third one, that is aerobic respiration. Take a screenshot. We will continue. Right? The two crucial steps of aerobic respiration are, now we know one step is TCA cycle, other step is ETS, electron transport chain. So the third one is aerobic respiration. If it has to do aerobic respiration, so the two crucial events are, so in NCERT they have given the first step is aerobic respiration means complete oxidation of pyruvic acid. Stepwise we have to remove hydrogen molecules. When you remove hydrogen molecules, C and O will be left out. It's called carbon dioxide. Complete oxidation of pyruvate by stepwise removal of hydrogens to make up carbon dioxide molecules. That's the first step. The two crucial events are complete oxidation of pyruvic acid, right? How do we do? By stepwise removal of hydrogen atoms. Step by step we will remove all the hydrogen atoms, leaving 3 molecules of CO2. By removing all the hydrogen atoms, when you remove, what happens? Leaving 3 molecules of CO2. Pyruvic acid, which is a 3-carbon acid. In this, if you remove all the hydrogens, first carbon, second carbon, third carbon, 3 molecules of CO2 happens. What is this called as? This is called as TCA cycle. What is this called as? This is called TCA cycle. Where does the TCA cycle occur? It occurs in the mitochondrial matrix. This is the first step of TCA cycle, aerobic respiration. The second process is passing on the electrons from the hydrogen. From the hydrogens you are removing the electrons, right? Passing on the electrons removed as part of hydrogen to O2. Oxygen is waiting to take the hydrogens, to take the electrons. So that's the second step, electron transport system. Passing off the electrons which are part of hydrogen to electron transport system. We are giving them to electron transport system and finally to oxygen. How are you giving? To whom are you giving? We are giving finally to oxygen. When we are giving off those hydrogens to oxygen, electrons to oxygen, then there will be simultaneous synthesis of ATP. When the electrons are moving from one carrier to another carrier, proton gradient will be built up that helps in synthesizing ATP. With simultaneous synthesis of ATP and simultaneous synthesis of ATP. This is the second step of aerobic respiration. What is this called as? This is called as electron transport system. And where does electron transport system occurs? On the inner mitochondrial membrane. ETS occurs in the inner mitochondrial membrane. One step occurs in the mitochondrial matrix. The next step occurs in the mitochondrial membrane. Understood? Now we will see what happens to the pyruvic acid if it enters aerobic respiration. Now when we start with pyruvic acid, what is pyruvic acid? A 3-carbon acid which is the end product of glycolysis. Where is it formed? It is formed in cytoplasm. Now the pyruvic acid comes to mitochondria. The pyruvic acid comes to mitochondria, and this pyruvic acid undergoes oxidative decarboxylation in the mitochondria. In the mitochondria, the pyruvic acid undergoes a step. What is that called? Oxidative decarboxylation. Oxidation also happens, decarboxylation also happens. Oxidation 1, we have seen it in the 6th reaction of glycolysis. Now that means this is oxidation 2 then. Decarboxylation in glycolysis, we did not see any decarboxylation. First decarboxylation we are seeing here. That means it is decarboxylation 1. Oxidation 2, decarboxylation 1, mediated by an enzyme complex which is called as, this reaction is mediated by an enzyme called pyruvate. Pyruvate dehydrogenase complex it is, okay. We can call it as Pdh complex. It is mediated by 3 enzymes and 6 cofactors. It is a shopping complex like thing in which many shops will be there. So Pdh complex contains 3 enzymes and 6 cofactors. We'll see what are the 3 enzymes and what are the 6 cofactors, right? So we'll write on this side 3 enzymes and 6 cofactors, right? So the first enzyme, since it is undergoing oxidation, it is called as dehydrogenase. The first enzyme will be dehydrogenase. Removal of hydrogens is oxidation reaction. Second enzyme will be decarboxylase. And since a 3-carbon pyruvic acid gets converted to a 2-carbon compound called acetyl-CoA. It is called as trans-acetylase. What is the third enzyme? It is called trans-acetylase. Oxidative reaction, so dehydrogenase. Decarboxylation reaction, so decarboxylase. Acetyl group is transferred, so trans-acetylase. These are the three enzymes which are present in Pdh complex. The 6 cofactors are, the first cofactor is TPP. The second cofactor is NAD. The third cofactor is FAD. The fourth cofactor is lipoic acid. The next cofactor is coenzyme A. The last cofactor is Mg2 plus 1, 2, 3, 4, 5, 6. 6 cofactors and 3 enzymes are there in this complex, which will convert a 3-carbon acid into 2-carbon compound called acetyl-CoA, which involves oxidation. Oxidation means NAD will come, take the hydrogens, gets converted to NADHH plus. For 2 pyruvates, 2 NADs will come, 2 NADHH plus is made. Decarboxylation means 2 carbon dioxides are released out. So coenzyme A is added. So acetyl group will take the coenzyme A and it will become acetyl coenzyme A. This reaction is called as link reaction because it is neither the part of glycolysis nor the part of next coming Krebs cycle. This is link reaction, oxidative decarboxylation mediated by Pdh complex. Take a screenshot, then we will get into Krebs cycle. Yes, now we are going to start Krebs cycle with the acetyl-CoA, which is a 2-carbon compound. Now we have seen that the Krebs cycle, TCA cycle, occurs in the mitochondrial matrix. Link reaction also occurs in the mitochondrial matrix only. Now we are going to start TCA cycle. We are going to start TCA cycle with the 2 molecules of acetyl-CoA. It is 2-carbon compound. This acetyl-CoA, it combines with 2 molecules of oxaloacetic acid, which is called OAA. So 2-carbon compound combines with a 4-carbon acid. The 4-carbon acid is oxaloacetic acid. Now it takes 2 water molecules also. The first reaction of this cycle is called as condensation reaction. What is the first reaction? Condensation. Condensing a 2-carbon compound with a 4-carbon compound. The enzyme will be called as citrate synthase. When 2-carbon combines with 4-carbon, you will get a 6-carbon compound which is called as citric acid. 2 molecules of citric acid are formed. Since citric acid is formed, the enzyme is called citrate synthase. The enzyme is called citrate synthase, and since the first product formed in the cycle is citric acid, it is called citric acid cycle. The citric acid cycle is a 6-carbon compound which is having 1, 2, 3 first carboxylic group, second carboxylic group, third carboxylic group. Citric acid is having 3 acid groups, so it is called tricarboxylic acid cycle. That is how the cycle got a name called tricarboxylic acid cycle, and this cycle was designed by Hans Krebs, so it is called as Krebs cycle. This cycle is also called Krebs cycle. Where does the Krebs cycle happen? In the mitochondrial matrix. What is the first reaction? Condensation, citrate synthase, citric acid is formed. Now this citric acid gets isomerized to cis-aconitic acid by losing water molecule. The cis-aconitic acid takes back the water molecule and get converted to isocitric acid. Means you can clearly see here that citric acid got converted to isocitric acid. Means 2 molecules to 2 molecules. So that means the second reaction is called as isomerization reaction. So the second reaction is called isomerization reaction. Enzyme will be called as cis-aconitase. Enzyme is called cis-aconitase is an enzyme. Removing water, giving back the water, means we are rearranging the groups, right? Now the third reaction will be isocitric acid undergoes oxidation. Isocitric acid undergoes oxidation. Oxidation 3 it will be, because oxidation 1 in glycolysis, 2 in link reaction, this is oxidation 3. And whenever oxidation happens, NAD will come, take the hydrogens, becomes NADHH plus. So we can write here that 2 molecules of NAD will come, take the hydrogen, becomes NADHH plus. Whenever HH plus is made, the enzyme will be DH. So the enzyme will be IDH, isocitrate dehydrogenase. Enzyme is IDH. Call it as isocitrate dehydrogenase is an enzyme. Now we are going to get an acid which is called oxalosuccinic acid. So 2 molecules of oxalosuccinic acid are formed. Isocitric acid is also 6-carbon acid. Oxalosuccinic acid is also 6-carbon acid. Now the next reaction is called as decarboxylation. So that means from 6-carbon compound, when you remove CO2, it will become a 5-carbon compound. So reaction number 4 is decarboxylation. 1 we have seen in the link reaction, so this is supposed to be decarboxylation 2. The enzyme will be called as oxalosuccino decarboxylase. The enzyme is called as oxalosuccino decarboxylase. And remove carbon dioxide molecules. Then a 5-carbon acid will be formed, which is called as alpha ketoglutaric acid, which is a 5-carbon compound. Next reaction will be alpha ketoglutaric acid gets converted to succinyl coenzyme A, which is a 4-carbon compound. This reaction is a replica of link reaction, means oxidative decarboxylation. So reaction number 5, oxidation also happens, decarboxylation also happens. Oxidation number 4, decarboxylation number 3, there it was Pdh complex, here it will be KDH complex, alpha ketoglutarate dehydrogenase complex. Since it is oxidation, NAD will come, NADHH plus will be made. Since it is decarboxylation, 2 CO2s will go off. So in the 5th reaction, oxidative decarboxylation, alpha ketoglutaric acid 5-carbon acid gets converted to a 4-carbon acid called succinyl coenzyme A. Enzyme is Pdh complex, same as KDH complex, same as Pdh complex. Now next reaction will be reaction number 6. Reaction number 6, where succinyl CoA gets converted to 2 molecules of succinic acid, which is a 5-carbon compound, that is 4. So this also will be 4. Just we are removing coenzyme A here. So when we are removing COASH, this bond will break up. That is high energy bond. When you break that high energy bond, so that high energy will combine with GDP with PI to make GTP. This GTP will give the phosphate to ADP, then you will get ATP. So for two ATP molecules, direct ATP is made in this reaction. When ATP is made, the reaction name will be phosphorylation. Substrate is giving the phosphate, so it is SLP, substrate level phosphorylation 3. So the 6th reaction is called as substrate level phosphorylation 3. 1 and 2 we saw in glycolysis. The name of the enzyme will be succinyl. S is there. Thiophosphate is added. Kinase. The name of the 6th enzyme will be succinyl thiokinase. Right? So the next reaction of Krebs cycle is conversion of succinic acid to 2 molecules of fumaric acid. Conversion of succinic acid to fumaric acid. This is also 4-carbon compound. This is also 4-carbon compound. Now when succinic acid is getting converted to fumaric acid, hydrogens are released. The hydrogens are taken by FAD this time. It gets converted to FADH2. H2 is formed, means oxidation. So oxidation number 4 you gave here. So this will be oxidation number 5. So reaction number 7 is oxidation 5. Enzyme, wherever H2s are made, SDH. So succinate dehydrogenase is an enzyme. Succinate dehydrogenase is an enzyme. We studied this in competitive inhibition in biomolecules chapter. Succinic acid is a substrate, fumaric acid is a product, malonate is a competitive inhibitor. Right? This is very important concept, right? Now, 2 molecules of fumaric acid gets converted to 2 molecules of malic acid, which is also a 4-carbon acid. So, in this reaction, 2 water molecules are added. Since water molecules are added, the reaction is called as hydration. So, reaction number 8 is hydration. The enzyme will be called as fumarase. The last reaction is conversion of malic acid into oxaloacetic acid. Reaction number 9, conversion of 4-carbon compound malic acid into a 4-carbon acid called oxaloacetic acid. Now, here what happens, again NADHH plus will be made. Means oxidation it is. NADs will come, take the hydrogens from malic acid, gets converted to 2 molecules of NADHH plus. When NADHH plus are made, so then the reaction is called oxidation only, last oxidation. So oxidation 6 it is, and enzyme will be MDH, malate dehydrogenase this time. This is called as Krebs cycle, right? So Krebs cycle, altogether 9 steps here. Now how to remember, I'll give you a code. The code is OK, simple for me is the code. OK, simple for me. See, citric acid, the starting compound we remember. Isocitric acid also we remember. We don't have doubt in these two. After that only we have doubt. Who will come next? Oxalosuccinic acid, alpha-ketogluteric acid, succinic acid, like that. So we made a code. OK, simple for me. O stands for oxalosuccinic acid. Here it is. After O, K will come. That means an acid with K will come. That is alpha-ketogluteric acid. Here it is. After K, acid with S will come. Succinyl-CoA and succinic acid will come. After S, an acid with F will come. That is fumaric acid. After F, an acid with M will come. That is malic acid. After M, come back again. An acid with O again. It is oxaloacetic acid will come. So like this, if you remember, it will be easy for you to study Krebs cycle. Right? So what is this called? This is Krebs cycle. Take a screenshot, we'll continue. Right? Understood. Now if we have to see the respiratory balance sheet. If we have to see the respiratory balance sheet, so we need to see how many NADHH plus are formed, how many FADH2s are formed, how many ATPs are formed. So 2 ATPs in glycolysis, the net yield of ATP in glycolysis 2 ATP. 2 ATPs here, so 2 plus 2, 4 ATPs. How many NADH2s? 2 NADH2s in the 6th reaction of glycolysis. 2 NADH2s in link reaction. 2 NADH2s here. 2 NADH2s here. So 8, right? Yeah. here also, right? So 2 NADH2s in glycolysis, 2 NADH2s in link reaction, 2 NADH2s here, 2 NADH2s here, and 2 NADH2s here. So 10. Total 5 2s are 10 NADH2s are formed. How many FADH2s are formed? Only 2 FADH2s are formed here. So when we talk about respiratory balance sheet, we need to remember these reactions. Okay. So let's go to respiratory balance sheet. Before going to respiratory balance sheet, we can finish off electron transport chain. Then we will talk about respiratory balance sheet. Now what is the second part of aerobic respiration? That is electron transport chain. Now whatever the reduced equivalents, means NADH2, FADH2s which are formed throughout till here, will enter electron transport chain. Electron transport chain, where it is located? On the inner mitochondrial membrane. Electron transport system. So electron transport system comprises of 4 electron carrier complexes. Where are they located? They are located on the inner mitochondrial membrane. We will draw the mitochondrial membrane. So this, what is this? This is inner mitochondrial membrane. And what is this side called? It is matrix. And what is this? This is a mitochondrial space. Perimitochondrial space. Right? So this is the outer membrane and this is the inner membrane. So this is the space. I told you electron transport system, it comprises of 4 electron complexes. So complex 1, complex 2, complex 3 and complex 4. If we have to see complex 1, so this is complex 1. Complex 1 is called as NADH dehydrogenase complex. So this is complex 1. It is called as NADH dehydrogenase complex. Now this NADH dehydrogenase complex, at this complex NADH2s which are reduced in glycolysis, which are reduced in link reaction, which are reduced in Krebs cycle will enter here and they get oxidized to NAD. The NADHH plus will come and they get oxidized, giving the electrons. Who will take the electrons? The electrons are taken by FMN. FMN will pass on the electrons to FES, iron sulfur protein. Now from iron sulfur protein, the electrons will come out of complex 1. Now when the electrons are coming out, when they are transporting through the electron transport carriers, protons are released out. The protons are released into the space. At this point, if you see from complex 1, 4 protons are accumulated in the space. Right? Now the electrons from here will now come to ubiquinone. UQ is ubiquinone. After taking the electrons and protons, ubiquinone becomes UQH2, reduced ubiquinol. Now the electrons of ubiquinol will be given to complex 3. Now the complex 3 is called as cytochrome B-C1 complex. So what is this complex called? This is complex 3. Now complex 3 is called as cytochrome B-C1 complex. Now it takes the electrons from here. Cytochrome B will take the electrons. It will pass on to FES protein. From there the electrons will go to cytochrome C1. Now from C1 the electrons will come to a mobile carrier. Now what is the name of the mobile carrier which is present here? It is cytochrome C. The mobile carrier which is present here is called cytochrome C, right? It gives the electrons. Now here also from complex 3, 4 protons are released from the matrix into the space. So at this point also, so we have to tell 4 protons gets accumulated in the space. Now when we have to talk about complex 2, now complex 2 is present here. Now the complex 2, what is it called? Complex 2 is called as Pdh complex, succinate dehydrogenase complex. Now when succinic acid is getting converted to fumaric acid, FADH2 is formed. That FAD will be here. FAD will pass the electrons to iron sulfur protein. From iron sulfur protein, again the electrons will go to UQ. UQ takes the electrons, it will become UQH2. It also passes the electrons to cytochrome B. So electrons, you can see from complex 1 they are going to complex 3. From complex 2 also they are going to complex 3 via ubiquinone. From complex 1 to complex 3 via ubiquinone. From complex 2 to complex 3 via ubiquinone. From this point also we can tell the 4 protons are released. From here also 4 protons, from here also 4 protons will be released. And the electrons will pass the same way and they will come to cytochrome C. From cytochrome C, the electrons are now taken by complex. The electrons are taken by complex 4. Now the complex 4, what is it called? Complex 4 is called as, we'll write, complex 4 is called as cytochrome C oxidase complex. The cytochrome C oxidase complex contains copper A. Then, so the electrons from cytochrome C will come to copper A. From copper A, the electrons will come to cytochrome A. From there to cytochrome A3. From there to copper B. So you can see how the electrons are getting transported here. From NAD to FMN, from FMN to iron sulfur, from there to a mobile carrier. It is not the part of this complex, not the part of this complex. So ubiquinone is a mobile carrier. It takes the electrons and protons, reduced from a ubiquinone becomes ubiquinol. It passes the electrons to complex 3. Complex 3 is called a cytochrome BC1 complex, which consists of cytochrome B, iron sulfur protein, cytochrome C1. From there the electrons will come to another mobile carrier which is located on the outer side. It is located on the outer side of the inner mitochondrial membrane, and it passes the electrons to complex 4, which is called cytochrome C oxidase, which contains copper A, cytochrome A3 and copper B. At this point also 2 protons will come into the space. So totally if you see 4 protons accumulated here, 4 protons accumulated here and 2 protons accumulated here. Now all these protons will build up a proton gradient. They will build up a proton gradient. Now these protons will try to cross. The protons which are more here, protons which are less here, they will try to cross across the membrane with the help of complex 5. Now the complex 5, what is it called? Complex 5, it is not part of electron transport chain. Complex 5 is called ATP synthase complex. It contains 2 subunits. The first subunit is a transmembrane protein. It is called F0. The second subunit is called F1, which is catalytic. F0, which is a transmembrane protein, it is embedded in the membrane. F1, which is catalytic, it's a peripheral protein, it is located towards the matrix. So F0, what it will do? F0 helps in pumping the protons. F0 helps in pumping the protons. For excuse me. For every 2 protons which are pumped from the space to the matrix, 1 ATP will be synthesized. one ATP will be synthesized. Synthesizing ATP by breaking the proton gradient, by passing the protons from F0 and F1 is called as chemiosmotic hypothesis. Who explained chemiosmotic hypothesis? What is this called? This is called chemiosmotic hypothesis. Chemiosmotic hypothesis explains the synthesis of ATP. Now the synthesis of ATP by oxidizing NADH2 to NAD, by oxidizing FADH2 to FAD, it's called oxidative phosphorylation. Synthesis of ATP by oxidizing the compounds is called oxidative phosphorylation. This concept of chemiosmotic hypothesis was explained by a scientist called Peter Michel. Understood? For every one NADH2 which is oxidized, it will make 3 ATP molecules. For every FADH2 which is oxidized, it will make 2 ATP molecules. Since NADH2 starts from complex 1 onwards, more protons gets accumulated here. For every NADH2 which is oxidized, it will give 3 ATP molecules. For every 1 FADH2 oxidized, FADH2 means from here only. That means we should ignore these 4 protons. So 4 and 2 only. For every FADH2 oxidized, only 2 ATPs will be made. Take a screenshot. We'll talk about the respiratory balance sheet now. Yeah, electron transport system also over. Now when we talk about the respiratory balance sheet, means when one glucose is oxidized to 6 CO2 and 6 water molecules, how many ATPs are accumulated? That's called respiratory balance sheet. Respiratory balance sheet. Now we told for every NADH2 which is oxidized to NAD via electron transport system, it makes 3 ATP. For every FADH2 which is oxidized to FAD through ETS, it makes 2 ATP. When we calculate the energy, first we'll go with glycolysis. In glycolysis, in reaction number 6, when phosphoglyceraldehyde got converted to 1,3-bis-phosphoglyceric acid, then 2 NADH2s are made. Now each NADH2 3 ATPs, so 2 3s are 6. 2 into 3, 6 ATPs. Now reaction number 7. In reaction number 7, when 1,3-bis-phosphoglyceric acid got converted to 3-phosphoglyceric acid, so ATPs are made, right? How many ATPs? 2 ATPs are made. So then 2 ATPs. Now coming to link reaction, all that we are just summarizing, we have seen all these things. In link reaction, when pyruvic acid, when 2 pyruvic acids gets converted to 2 molecules of acetyl-CoA, 2 molecules of NADH2s are made. Each NADH2 will give 3 ATP. So 2 into 3, 6 ATP in this reaction. Then next, when we talk about TCA cycle, in TCA cycle, when oxalosuccinic acid, when isocitric acid gets converted to oxalosuccinic acid, right? There 2 molecules of NADH2s are made. So 2 into 3, 6 ATPs. The immediate reaction when we take like KGA getting converted to succinyl-CoA. In this reaction again 2 NADH2s are made. So 2 into 3, 6 ATPs. Substrate level phosphorylation again in TCA cycle. When succinyl-CoA gets converted to succinic acid, so 2 ATPs are made. The next one, it's again in TCA cycle. So what is that reaction? When succinic acid gets converted to fumaric acid, 2 FADH2s are made. Each FAD will give 2 ATP. 2 into 2, 4 ATP. Then again in the TCA cycle, the last one where malic acid gets converted to oxaloacetic acid. So 2 NADH2s are made. So 2 into 3, 6 ATP. Now calculate. So 6 plus 2, 8. 8 plus 6, 14. 14 plus 6, 20. 26, 28, 30. 28, then 32 plus 36, 38 ATP. So when we calculate, so how many, we get 2 ATPs we are getting here. One more thing we need to write. What is that? Reaction number 10. I forgot to write. Reaction number 10 of glycolysis. In reaction number 10 of glycolysis, where phosphoenol pyruvate gets converted to pyruvate, then 2 ATPs, right? So when you take that 2 ATPs, total 40 ATPs will come. When you calculate all these ATPs, 40 ATPs will come. Out of 40 ATPs, in the first reaction and third reaction, we use 2 ATPs. So 2 ATPs consumed. So if you minus that 2 ATPs, 40 minus 2, 38 ATPs will be the net ATPs. So this calculation when you make, so this is a beauty to calculate, but for this you have to make certain assumptions. So we have to think that person is taking only carbohydrate diet and the steps are going in sequence: glycolysis, Krebs cycle, electron transport chain. The intermediates are not deviating. When we make all these assumptions, then only you can calculate this balance sheet of 38 ATP molecules. Take a screenshot, we'll continue. Right. Now when we talk about amphibolic pathway, till now we are telling respiration is catabolic since we are breaking food materials. But when we see, it's not only for breakdown of food materials, it is also used for making. So when we see glucose, carbohydrates, they are broken down to glucose. Glucose is broken to fructose. Fructose is broken down to phosphoglyceraldehyde and dihydroxyacetone phosphate. Then this gets converted to this and then we get pyruvic acid. This pyruvic acid enters Krebs cycle and then water molecule and carbon dioxides are released out, right? So this we have seen. Carbohydrates first they get converted to glucose. This is glycolysis. Then glycolysis, they make pyruvic acid and we have to tell pyruvic acid gets converted to acetyl-CoA also. Then from acetyl-CoA, Krebs cycle will happen. So if we are taking fats as the respiratory substrates, fats are first broken down to fatty acids. Now these fatty acids are long chain aliphatic compounds. When we break fatty acids, so then we get 2 carbons each. Then those 2 carbons are called acetyl-CoAs. Means fatty acids also enter the pathway at acetyl-CoA level, right? So then if you take protein, proteins are broken down to amino acids. Amino acids, if you do deamination, remove amino group, they can become pyruvic acid. They can become pyruvic acid or they can become any of the intermediates of Krebs cycle. That means not only the carbohydrates, proteins and fats are also entering the pathway. And so when we eat too much of chocolates, then we become fat because the carbohydrates can make fat. When we take too much of protein also, that also enters. Means all the 3 pathways are interconnected at this junction, which is part of respiration. Means the respiratory pathway is not completely, it's not purely catabolic, it is anabolic also. So carbohydrate, excess carbohydrate is to make fat. So that's why we call respiration as amphibolic pathway. What do we call it as? Amphibolic pathway. Amphibolic pathway means it is used for breakdown also. and it is used for synthesis also. For synthesizing fat, for synthesizing protein, we can use the same pathway. That's why it is called amphibolic pathway. Right, children? Now take a screenshot. We'll talk about the last topic, that is RQ, respiratory coefficient. Right. So what is RQ? It is called respiratory coefficient. Now, how can you calculate the respiratory coefficient? What is the value of respiratory coefficient? Volume of carbon dioxide evolved to the volume of oxygen consumed. So, RQ value is, the formula is volume of carbon dioxide evolved to the volume of oxygen consumed. Now, if you take glucose, for carbohydrates, if we go, C6H12O6 plus 6O2 gives rise to 6 CO2 plus 6 water molecules. That means 6 CO2 by 6 O2, right? 6 CO2 by 6 O2. So the RQ value will be 1. For carbohydrates, the RQ value will be 1. For fats, if we go, for example, take tripalmitic acid. Now, tripalmitic acid molecular formula will be C51. It is C51H98O6. So, if we take this molecule, 2 molecules if you take, how many oxygen molecules? Means 145 oxygen molecules it will take. It takes 145 oxygen molecules to release 102 carbon dioxides only. Then the formula will go 102 CO2 by 145 O2. The denominator is more. If you calculate it, it will get 0.7. What is the value? When we simplify, we will get 0.7. That means fats will have an RQ value of 7. In the same manner, if we go with protein calculation, so it is difficult to remember. In NCERT, they have given the RQ value of protein will be 0.9. For carbohydrate, it is 0.1. For fat, it is 0.7. For protein, it is 0.9. And for organic acids, the RQ value will be more than 1. And if we go with anaerobic respiration, in anaerobic respiration, only CO2 will be there. O2s are not there. The RQ value for anaerobic respiration will be infinity. Remember this, children. RQ of carbohydrate 1, fat 0.7, protein 0.9, organic acids more than 1, and for anaerobic respiration it is infinity. What is the significance of RQ? So depending on the value, we can tell the nature of respiratory substrate. We can tell the nature of the respiratory substrate, right? So with this, we completed the chapter number 14, respiration in plants. In the next lecture, we'll try to see the MCQs which have been asked in NEET examination from this chapter. Thank you.